So today I'll be talking a lot about our methods for both learning and characterizing dynamics from actually various kinds of biological data, but most of these data sets are at the cellular level. So my lab generally tries to see what kind of biological insights we can learn from various sources of biological data. We've worked a lot with single cell RNA sequencing. We've also worked with molecular and even non-coding RNA, but also increasingly signaling and activity data like fMRI or calcium signals. And the big question is, a lot of these data sets are pertaining to this awesome complex system that's called a cell. And what can we really learn about cells from all of the data that's being generated? Because there's far more data now than any scientist can really pour over. And that's really the theme we're trying to tackle in my lab. How do we use AI to really extract these kinds of insights? So there's a lot to do in these data settings. So in the past, my lab has worked a lot on denoising data, visualizing dimensionality reduction, multimodal, multi-sample alignment. In this talk, I'll be focusing on dynamics. So there's actually sort of two goals with respect to dynamics. The first one is that many of our data types, including single cell RNA sequencing, which is one of the most informative data types, as well as other single cell modalities like CyTOF and ATAC-seq, give you data that is static snapshot. So that means that you have time points, and these are often spaced far apart, even if you have 4, 5, 6 time points, which is kind of the extent of the timing in most of the data sets today. So they could be days apart, weeks apart, and there's a lot of biology that really happens in between, because those biological entities are actually continuous entities that exist continuously in time. So theme in our work since about 2020 has been, can we interpolate what happens in between? So can we interpolate the distribution at time points we didn't measure, but can we also understand what would have happened to a single cell had it been followed in time using some kind of technology that we don't yet have? And so this is inferring the dynamics from static data. The second kind of goal we have with dynamics is characterizing the dynamics. So this is from a recent paper we had called TFATE in Nature Computational Science. And what you see here are different brain state trajectories from different areas of the brain. So these are very complex dynamics. So how can we classify or learn something about the dynamics that's going on? So are these dynamics different between people who process differently in the brain, for example, people who have autism or schizophrenia? So that's a matter of actually coming up with descriptors and quantifying meaningfully differences between dynamics. So I'll talk a little bit about both with a little bit more emphasis on the first one. So before I get into that, I'm going to explain the first part of my talk, which says deep geometric. And a overriding theme in our lab that has been very helpful in tackling all kinds of biomedical challenges is this kind of hypothesis that the data has geometry and that it has manifold geometry. So the idea that scientific data is sampled from a manifold and then perhaps perturbed a little bit with noise. This allows us to learn the shape of the data, which is very important for learning dynamics as well, because if you're going to infer dynamics from data you don't have, you want to understand what is the ground space where these cells are moving or these brain state trajectories are evolving. And the proof is a little bit in the pudding. A lot of our methods have done very well once they've taken data geometry into account. One of the reasons why this is a good assumption is because biological data has high ambient dimension, but there's an underlying latent space that's much lower dimensional. And biological entities tend to move in gradation. So you will see this, you can learn the shape, and the shape will be filled out if you have enough data, so to speak. A common theme we use for manifold learning is using the concept of an affinity graph. So here you have, for example, in single cell RNA sequencing, cells by genes. We can turn that into a weighted graph or an affinity matrix by calculating distances between these feature vectors, a Euclidean distance, and then pushing that through some kind of a kernel function that inverts it into an affinity. And some of these have sharp drop off, which emphasize local connections. That means you're not assuming anything about global distances, but rather you're happy to walk along the data structure to discover this shape. And one tool that we use in addition to the affinity matrix is the diffusion operator, which is sort of a normalization of the affinity matrix that allows you to actually conduct walks of t-steps via powering. And that gives you long paths along the manifold and allows you to learn the shape of the manifold. The diffusion operator has also been useful in other data sets for us, like molecular data sets, for example. A good way of understanding the structure of a molecule is to put signals and see how they diffuse on the molecular graph, and I won't get a chance to talk about that. Recently, we've also discovered that this kind of diffusion operator is a discretization of a heat operator. And there's very well-known connections to manifold distances to the heat operator that go all the way back to 1967 Vardhan's formula. And recently, we've been invoking that more to directly even use heat. The diffusion operator we use actually came from one of my colleagues, Rafi Koifman, almost 20 years ago now, when they discussed their diffusion maps embedding. And here, the eigenvectors of the diffusion operator created a new space. We don't often use this space. We use different spaces like our fate space. But nevertheless, something that's very important about this eigenspace is that it separates out structures in the data, as well as allows you to decompose features on those data graphs like genes into low and high frequency components such that you can denoise them. So these are a little bit of the tools that I use. And based on these tools, we've developed a lot of methods. You may have heard of magic or fate or meld or some of these. I won't be talking about those today, but I will refer to them. Today, I'll be talking about how to bring together data geometry and some topology together with deep learning to really learn and characterize dynamics in data. So I'm going to start with a method that we published in NeurIPS this past December called MIOFlow, or manifold interpolating flows. So this is one of our latest models for inferring dynamics from static snapshot data. I'm just going to, and this is kind of a schematic of the network. And we're going to talk a little bit about how each of these components work. So the idea here is that we could have time course data in single cell. This is embryoid body data that we generated as part of our FATE paper, stem cells differentiating eventually into different lineages. And as opposed to previous methods, which are useful, so there's other methods that have been looking at dynamics like RNA velocity that gives instantaneous arrows. There's Waddington OT, which gives couplings. We wanted to do something different. We wanted to actually learn continuous dynamics and eventually even make these dynamics interpretable. So for this purpose, we first regularized an autoencoder with a multi-scale diffusion distance, which is similar to the distance you get in a diffusion map, except it's at many, many different scales. And it turns out this kind of multi-scale diffusion distance actually gives you an optimal transport between data sets. If you look at it between data sets, it's a dual form of an optimal transport distance. So we proved this in this paper called diffusion earth movers distance. So the idea is you can combine many different data sets into one graph, you create an indicator signal, and you diffuse to multiple scales. And when you take the difference between those scales, you'll get optimal transport distances between those populations. So that space turns out to be a very useful space. In this space, you can get an earth mover's distance just by subtracting densities at different scales. And the ground distance is a manifold distance. So MIOFlow makes use of that by embedding data into the space of those features. And then it runs a neural ODE solver. So this is a neural ODE. This part is a neural network. The neural network produces the derivative, and this derivative is integrated out with an ODE solver. And you could even use an SDE solver. So what that means is the neural network is actually learning continuous dynamics. And what our penalty is an optimal transport penalty. And so diffusion EMD results in a differentiable optimal transport penalty. So our neural network for each cell learns the derivative of the cellular state. In this latent space, the ODE solver will give you what that cell would be at a future time point. And at that time point, we give a population level optimal transport loss. And that's what allows MioFlow to infer continuous dynamics. In addition, we penalize the magnitude of the derivative. And it turns out that that is important for performing what's called dynamic optimal transport. And we're able to prove that if we have this penalty in place, that MioFlow gives a dynamic optimal transport using this flow from the neural network, which is regularized. So just to give you a little bit of idea about dynamic optimal transport sort of gives you the most efficient path a cell could take, because cells are energy constrained entities and they would want to take efficient paths between states. So dynamic optimal transport takes points exactly where they want to go, rather than kind of sliding them through meandering paths. And we've seen before in our trajectory net paper that even continuous normalizing flows, when you add that energy regularization, give you something closer to dynamic optimal transport. We're able to add other penalties, such as unbalanced transport for cell death and growth, as well as even utilize other information like the RNA velocity that I mentioned. If we have it on our data set, or if it's reliable on our data set, we can constrain the paths where RNA velocity is available to match with it. But you can see there's gaps in the data where it might not be available. So we've tested MioFlow against Schrodinger bridge, diffusion's Schrodinger bridge, and trajectory net. And you see MioFlow can follow curvy paths pretty well in this artificial flower petal data set. On the stem cell data set I initially showed you, it pushes data along this optimal transport path, and in its wake, it leaves these single cell trails. So you recover at the single cell resolution the trajectories of the cell. So each cell has a trajectory as a result of this. This is what makes it different than something like pseudotime. Pseudotime gives you one trajectory. Now we have a trajectory for every single cell. So we've been using that on a very exciting system that we finally submitted the paper on and put on bioRxiv. And it's a system of triple negative breast cancer. So in triple negative breast cancer, there's processes called EMT and MET that endow cells with plasticity. And this plasticity is what has been known to allow cells to evade therapy, for example, chemotherapy. And in general, EMT is studied fairly well, but MET had not really been studied as well. So with our collaborator, Christine Schaefer, we grew these 3D mammosphere cultures that form these tumor-like entities. And we were focusing on sort of this transition where cells can transition spontaneously from a poorly aggressive state to a state where they can lead to metastasis. And in order to do this, we collected 5 time points of data, again, spaced apart by about a week. And from this data set, we initially ran trajectory net, and recently we ran MioFlow. And this gives us the single cell trajectories that you talk about. It turns out that this is an apoptotic population, and the trajectories we care about are over here. So one thing we were able to do is, if you take a look at this, because of our unbalanced transport, not every cell transitions. Some cells die, and you can look carefully to see which are the cells that create those secondary tumors and create additional tumor spheres. And we can characterize them by cell cycle, but also by a plethora of markers. And it turns out that this refines what's called the cancer stem cell signature for this particular cancer type. And this refined signature, we're able to test in the lab by actually putting the cells that express some of these markers in separate spheres and seeing that they create larger spheres more consistently. And you can read more about that in the paper. These single cell trajectories over time allow us to look at different types of trajectories. And it turns out there's actually 2 kinds of trajectories. One is tumor forming and one is non-tumor forming. And we can differentiate the dynamics between these trajectories. Because we have cell trajectories, we can have gene trajectories for these 2 cell fates, some of which we can validate in vivo, but not nearly as much data as we get in silico. We get gene trajectories for every single gene. So what we did was we ran Granger causality on these gene trajectories, looking at the genes that come up first, that come up later, that come up at the end. And we're able to cross-link that with a prior knowledge network to arrive at this large network. And in this large network, we find a hub where there are potential promising targets for this cancer type. In fact, AR was already under clinical trial by our collaborator who found it using a different method, but we see it in our network. We've honed in on these two and we've been testing this one, and it looks promising in mouse work so far. So when you knock down Ezra, you see a return of CHD1, which means it's stopping the EMT, and you see the same thing in this Western blot type of plot. And this is kind of where we're at this project, and that paper is under review, but you can check it out on bioRxiv. This work is unpublished. It's very recent. We just submitted it to NeurIPS, I think like a week and a half ago. So once we did that work with the cancer, what we realized was the networks that we built at the end of it are really very important. So we created another neural network that automatically infers that network and doesn't reference Granger causality, which we've had some problems with in some circumstances. So the way we did it was we trained a new neural network to match the output of MioFlow, but we put a graph, a regulatory graph, right inside that network. And so this is a graph ODE. The idea of it is that the genes, when they dynamically change their expression pattern, it's in response to altered interactions in the regulatory network or interactions that are taking place in the regulatory networks. And so can we create a generative dynamics that follows this principle? So this network is called Retini. This is a graph ODE network. And basically the graph ODE is trained to match dynamics, but using but the dynamics within the graph is generated using graph attention on its neighbors. So we can weaken and strengthen and even add edges using some unique attention mechanisms that we have. So here we have attention over time. So we can look at different time lags of a signal. And this again is an ODE, so it computes a derivative. And that attention, that we have spatial attention over neighbors. So we have space and time attention. And we also featured time lag between nodes. So we're looking at lagged values of neighbors in order to predict the next nodes. And we're also looking at autocorrelation of the signal. And the time attention itself can look through an interval of time. So this could support other things like hysteresis, which we tried in neuronal networks. So this is a attention network that gives a time varying graph. It gives a graph that evolves with time, much like regulatory networks do. It can support either single time point memoryless dynamics, or it can support hysteresis. And it outputs at each time an instantaneous derivative that is integrated. And the loss function is similar, except for we have a small neural network outside the initial derivative. And the loss function also features a regularization by a prior, so we could still take into account prior knowledge and enforce sparseness. So when we started working with this, we noticed that we could actually train this with perturbations. But interestingly, it's better to train with mild perturbations than heavy perturbations like drugs or CRISPR knockouts. Interference is better. And the reason is because if you have a small perturbation that's timed and it's applied, its effects propagate through the network. And if you propagate its effects through the network, you understand the structure of the network better. Whereas if you have a heavy perturbation, then it rewires the network and you don't gain insight into the original network. So we showed how we can actually strengthen this learning by including perturbations, which are basically disruptions to the signals at the node during training. So you have a node here, you disrupt it, and then you see its effects cascade through the network, and that aids in the training. We've tested it on several so far simulated methods, as well as some single cell RNA sequencing data. These are artificial dynamic simulations where we know the ground truth. The issue is with the real data, we don't always know the ground truth. This is a neuronal network of neurons, a simulator called NEST, and a gene regulatory network simulator called Sergio. So this is how we computationally validate it. And then we see how the ground truth network that was used in the simulator we see how similar our method is compared to the other methods. And this is with the gene regulatory network that was inferred using Sergio. So this paper is going to be on arXiv fairly shortly since we submitted it. So this is kind of where we're at. Our ongoing work is trying to incorporate spatial information into the graph that's inside Rotini in a couple of different ways by modeling cell-cell communication as well as conditioning on the microenvironment. I think I have a couple of minutes, so I'll quickly talk about characterizing dynamics. So this is a paper that recently came out from our lab together with the Valentina Greco lab in the Journal of Cell Biology. Here we had the dynamics like we would if we inferred it through Rotini. The question is, what can we do with the dynamics next? Can we understand something about the system? Can we put into machine learning? And so we were looking at a very interesting system. This is calcium signaling in epithelial cells. Cycling stem cells communicate via calcium signaling. And this is what the real data looks like. This is the real data generated by our collaborators. If you see this, you might think you're seeing random flashes. Like, do they have any meaning? It's not like neurons where people know what the calcium signals does. Is it random? Is there coordination? And this is why we needed a computational method to figure it out. And so we have a computational pipeline that we call GSTH. The first step is to create a graph, which is a nearest neighbor graph. Then we capture the signaling pattern in space by something called a scattering transform. Then we apply our new temporal fate method to it. And then finally, we capture the whole trajectory that we see using topological data analysis. Since I'm hurrying through this, I'll kind of do it quickly. This is the graph creation. Then you can treat the signals as a graph signal, and then you can diffuse those signals to different scales. And this leads to something called a geometric scattering transform, which is a multi-scale, multi-order diffusion wavelet transform on those signals. Diffusion wavelets are created just by differences between two scales of the diffusion operator that I previously showed. And these are chained into a deep multi-layered network, which we can make learnable again. So again, deep geometric. And now when you look at fate, you see that those flashes are not that random. They're actually following a very smooth trajectory. And in fact, if you disrupt something in the cellular system, the smoothness gets disrupted. So the way we saw that this was smooth was by taking each time snapshot and applying fate to it. Temporal fate is an improvement on fate where it has a temporal autocorrelation view in addition to the fate view. And so when you're looking at these random flashes, fate actually is able to draw out a very smooth trajectory that follows this shape. And then the topological data analysis gives us, this is Vietoris-Rips complex. This is a persistence diagram that featurizes it. And I don't have time to go through how that works, but what you can see here is this kind of smooth signaling sweeping through the tissue. Is everything okay? Is everything okay? Is unique to this these epithelial cells. If you looked at calcium signaling in a neuronal cortex, it's not that smooth at all. And when you disrupt the signaling in epithelial cells, you see disruption in the smoothness of it. And so this is kind of where I wanted to stop with that. We also were able to recover the effect of drugs and also recover parameters of other dynamic systems that we modeled with this. So most of this work is in the Journal of Cell Biology paper that I talked about. And finally, I just want to thank all the wonderful collaborators. I have Valentina Stem Cell Data, Christine Schaefer, who generated the cancer data, Guy, who's a frequent collaborator, and John Locke, who's an expert on imaging, and all my students and postdocs. Thank you.